Okay. Right. So some of us have been through a tutorial where we went over glycolysis and nicotine, adenine dinucleotide, and pyruvate, and mitochondrion, and cytosol, and all that stuff. I'm going to have to go through it again very quickly because it's very important that I have you guys along for this. So at the moment, what you're seeing on screen is on page 128 in your books. I need to speak about glycolysis because I want to cover first aspects of cellular respiration. It is in a different order to what's in your book, but unfortunately this has been thrust upon us. So for anybody who hasn't been here, what you need to know about glycolysis is, first of all, that it occurs in the cytoplasm. It's a cytoplasmic process. It does not occur in the mitochondrion. Second thing that you need to know about glycolysis is it is ubiquitous. It occurs in every single cell. It's the beginning of all forms of respiration. If oxygen is not present, glycolysis will be as far as it goes. But the end product of glycolysis, which is pyruvate, will be brought to the mitochondrion for further processes of respiration. Is that okay? So, we know that glycolysis, because of its name, is the splitting of sugar, and it's dividing a hexose, which has 6 carbons, into 2 trioses, or 2 3-carbon fragments. These 2 3-carbon fragments are known as pyruvate, and they will be what travels to the mitochondria. What else does glycolysis produce? It also produces 2 other things that we're interested in. As well as breaking down 1 glucose into 2 pyruvate, it also will give us 2 ATP. Now the ATP will be created by removing a phosphate from the sugar, which has been added during the process, and putting this onto ADP to form ATP. This will be done twice for every glucose that comes in. Now looking at it, it looks like it's been done 4 times in this diagram. Well, that's because it's only been done twice net. 2 go in, 4 come out. That is a gain of 2 net. Okay, the other thing that is important is we have created NADH. NADH is a product which has been produced in a redox reaction. A redox reaction, meaning the NAD plus becomes NADH as it gains 3 things. In its reduced state, it has now gained free energy. It has gained electrons, and it has gained protons. Is that okay? Now if you want the actual number of it, well, it will have gained 2 electrons and 1 proton. So NADH is a coenzyme. It's an organic molecule that works with a particular enzyme. This enzyme will oxidize sugars, stripping away energy, protons and electrons. And the electrons, protons and free energy will be transferred to this cofactor, or coenzyme, which becomes NADH. So it has 2 different forms. This is the reduced form, and this is the oxidized form. Okay. So respiration, it says here, is essential to all living things. It happens in every cell, and it begins, for our purposes, with glycolysis. It can be carried out on fats, proteins, but we're only interested in what happens when we apply it to glucose. Okay. So the process of breaking down glucose begins with glycolysis. We'll be talking a lot about ATP, and if you're not familiar with it, here is a picture of it. It is a ribonucleotide. So it's very similar to the kind of monomers that go into making RNA. Yeah. It contains a nitrogenous base up here, and that is called adenine. It's linked on to a 5-carbon or pentose sugar. In this case, it's called ribose. And then here it contains 3 phosphate groups bound onto carbon number 5. Is that okay? ATP can be thought of as a full battery, and ADP being a battery that has lost some of its charge. Good. So we know that hydrolysis, or removing, or dephosphorylation, or removing the phosphate group from ATP would be an exergonic process. In other words, if you strip off the last phosphate, you can release a very large amount of energy. So ATP itself contains a lot of energy. It wants to be released, but it's got a high activation energy, so it does not spontaneously break down in the cell. What does it need in order to be dephosphorylated? Something that could reduce this activation energy? Yeah, some sort of an enzyme. So it needs an enzyme to release the phosphate. What's the point in having an enzyme to release the phosphate? Well, that enzyme will be releasing the phosphate in order to do some other job. Two different reactions will be coupled, meaning that the endergonic job can be done because the enzyme is also stripping a phosphate from the ATP. Is that okay? In the same way that if we have an electronic device, it does two jobs. One, it acts as a computer, but also it acts as a battery emptier. You know what I mean? The only reason it empties batteries is so that it can provide us with the other useful function. The two are coupled. In that way, ATP serves as the fuel to power the process. What kind of work is ATP used for? Lots of different work in the cell. We'll mention many of these. It's not important to remember this list, but these are just examples of what kind of things needs to be done. Mechanical work, like we would see in muscles or for movement. Electrical or concentration work, I mean pumping, transporting things across membranes. Or synthetic. When you go into biosynthesis, you have anabolic pathways that build up. They also need energy to be input into the molecules you're going to be making. Is that all right? Wonderful. Okay, so what types of respiration are there? Well, first and foremost, it's broken down into anaerobic and aerobic. Anaerobic means takes place without the need for oxygen, but aerobic requires oxygen. We work on aerobic respiration, isn't it? And if our muscles are forced to carry out anaerobic respiration, it's very brief. And it's not sustainable, isn't that the word? Yeah, like we imagine we run from a standing position, we're relaxed, we start running after the bus, we get a pain in our side because our diaphragm is attempting to keep working even though the oxygen levels are falling. And so it produces a process, we'll look at it in a moment. So we've also met the words obligate and facultative before, haven't we? Obligate means I must do this, facultative means I can do this. So we used it for parasitism, and now we're applying it to aerobic respiration. We are obligate aerobes. We cannot do anaerobic respiration. Just because we can do it for 3 seconds as we run for a bus does not make us anaerobes. But an example of a classic facultative anaerobe would be yeast. Yeah, if you put yeast in normal situation, it will aerobically respire and be very happy to do that. But if it finds itself in anaerobic conditions, it will be able to carry out fairly sustainably a process of anaerobic respiration. Yeah, and we'll talk about that in a moment. So, but both anaerobic and aerobic begin with glycolysis. So glycolysis, whether it's an aerobic or an anaerobic organism, if you present them with glucose, the first thing they must do is glycolysis. As its name suggests, the sugar is being split. So splitting the sugar, hexose is going to become 2 3C sugars. In this process, we're going to create 2 ATP from ADP, and we're also going to convert 2 molecules of this coenzyme, NAD+, into NADH. Is that all right? So NADH will be released from glycolysis in 2 oxidation reactions, and also in 4 places we will get phosphorylation that comes from the sugar. And you can see the details of it coming from the sugar in a moment. We don't need that to expand. On we go. Okay, this is what NAD+ looks like. It's a terrible picture of it, but we don't need to know the structure of it. It's not a big deal. All it is a coenzyme in the body that is able to flip between an oxidized and a reduced state. When it's in its oxidized reduced state, it is poor. It lacks something. But when it's in its reduced state, it has gained it, isn't it? Reduction is gain. What has it gained? These 3 things. For our purposes, I want you to remember that redox reactions in cellular respiration, that which is reduced gains electrons, protons, and free energy. Yeah. Meaning that NADH is a very, very useful thing if you have oxygen. If you don't have oxygen, it's not very useful, and it can in fact become quite a problem. Because what does glycolysis need in order to proceed? It needs glucose, yeah. If you don't have glucose, it will stop. What about oxygen? Does it need oxygen to proceed? No. What else does it need to proceed? It needs a little bit of ATP ready to go for that input, yeah? And it will need some ADP to create ATP further on. And it has an absolute requirement for NAD+. If the NAD+ is not available, glycolysis will not be able to proceed. Is that okay? So if in my cell I convert all of my NAD plus to NADH, that sounds wonderful, doesn't it? I've got free energy in there, but unfortunately it would mean that glycolysis will stop if I don't have some way of getting rid of those electrons. And oxygen, as we know, loves electrons. That will be our solution to the problem of what do we do to get rid of the electrons. Yeah. Give them to oxygen. What happens when you throw protons and electrons at oxygen? Water. We get water. Yeah. Okay, so NAD+. So 2 complete hydrogen molecules will be stripped from the sugar by an enzyme called dehydrogenase. It's not vital that you remember the dehydrogenase. It would be useful if you could. But dehydrogenase enzymes are active in cellular respiration because they oxidize the sugar. If oxidation is loss, then we can say that these enzymes strip something away from the sugar. And this is quite intuitive because the sugar has arrived like an old car has come to the dismantling yard and they're going to take all the pieces of it off, aren't they? And hopefully there'll be nothing but scrap metal and products you can sell at the end. Yeah. What do we expect is going to be available or is going to be produced at the end of cellular respiration? Well, we're going to have some energy in the form of ATP, sure. But what about the raw materials? What will happen to the C6H12O6 that came in? Yeah, the carbons and the oxygens are going to be put into carbon dioxide. Yeah. And what's going to happen to that hydrogen that's been taken off? It's going to go with oxygen gas from the atmosphere to form water. Yeah. So hopefully at the end of at the end of cellular respiration, we'll have carbon dioxide and water, as well as lots more ATP that's been created. Is that okay? Yeah, wonderful. So you can go back and you can look over the details of the redox stuff there. But the main thing to remember is oxidation is loss, reduction is gain. What is being lost and gained in cellular respiration? 3 things. Protons, electrons, free energy. Yeah. Okay. Some things you should never say. Please never say NAD plus is reduced or NADH is reduced, because they're both true. NAD plus is reduced is true in the sense that NAD plus is reduced to become NADH. Also the opposite, what seems like the opposite, is also true. NADH is reduced, NAD plus is oxidized, meaning they are in the reduced state or in the oxidized state. So don't use the verb to be in this sentence. What should you say instead? You should say NAD plus becomes reduced to become NADH, or NADH is the reduced form, meaning it's in the reduced state. Yeah? Use a different verb so you don't make an ambiguous statement. Okay. We have mentioned before about the potential energy of electrons. We've said that some electrons have got more energy than other electrons in the same atom. Is that true? Depending on their distance from the nucleus? Yeah. Also, we also have seen that some atoms tend to hold electrons at greater energies than other atoms because of their electronegativity. Is that okay? Yeah. So what we're hoping for is to show that we bring in a sugar that carries quite high energy electrons, we transfer them to something that holds them at a lower energy level, but still pretty high, and then we hand them on to a whole series of different carriers. Every time we give them from one substance to another, it's another redox reaction because electrons and free energy are being transferred along. And then at the end of all of these series of redox reactions, it will eventually end up with water. And water, we know, oxygen in water holds the electrons very, very tightly indeed at a very low energy level. Yeah. So at every stage, as we take the electrons from the sugar to the oxygen, we should be releasing in controlled amounts of energy, which we can use at every stage to make ATP. Is that okay? A little bit like a ball bouncing down the stairs. Is that okay? Good. All right. So oxygen is very electronegative. We know that. It has a high affinity for electrons, meaning it wants them, and when it has them, it holds them tightly and closely. So oxygen often oxidizes other molecules. Is that okay? It draws towards itself electrons in particular. The transfer of electrons from sugar to NAD plus loses a little but very little energy. Is that all right? So we can transfer electrons from sugar to NADH. It's favorable, it's an exergonic reaction, but it doesn't lose too much. So they still retain lots of their energy. So that in that way, NADH is an energy store. It has free energy that is available to be used. It can reduce other things. Yeah? So So electrons will flow from NADH towards oxygen, releasing a lot of energy, and will move in a number of stages. And I'll show that in a diagram in a moment. Okay, if you want to look for any close details on glycolysis or to help you understand what's going on, these two sets of diagrams are wonderful. Pay particular attention to how we put ATP in and look what happens to the phosphates. At step number 1, what has happened to the phosphate? It's gone onto the substrate or onto the product, isn't it? Yeah. And again in step number 3, what's happened to the phosphate that came from ATP? It's gone onto the sugar as well, hasn't it? So now the sugar, which came in without any phosphates, has been phosphorylated twice. Yeah. Now in the magic step number 6, all kinds of wonderful things happen. First of all, there's a redox reaction in step number 6. It's occurring twice. So that's why we have 2Pi entering, 2 NAD plus being reduced. Why is it occurring twice? It's occurring twice now because that 6 carbon fragment we saw at step number 4 has been split into 2. Now, dihydroxyacetone phosphate does not go anywhere. The only thing that actually goes anywhere from this reaction is G3P, glyceraldehyde 3-phosphate. So what it means is for every glucose that comes in at step number 1, 2 G3P will exit at step number 6. Can everybody get their heads around this? For every 6 carbons that come in, 6 must leave. So for every single glucose that comes in, 2 G3P move out. And what's happening is the dihydroxyacetone over here, sorry, my apologies, the dihydroxyacetone is moving across by equilibrium. So as we remove the G3P, the dihydroxyacetone becomes G3P. They switch freely as an equilibrium reaction. Is that okay? So everything happening on the second page is happening twice. There should be a 2 on everything, isn't there? Including each molecule. The picture you're shown has got a 2 in the corner. Yeah, everything's happening twice. Okay, so what happens on number 6? Well, the magical step happens of a lot of energy is taken from G3P as it is converted. This energy is turned into phosphorylation and turned into a redox reaction where NADH is produced. Meaning that we end up with 2 individual molecules that both are bisphosphates. They've both got 2 phosphates on them. And then when we remove those 2 phosphates, we will be able to create ATP. Take home message, what do you need to remember? 2, 2, 2. Yeah? 2 pyruvate, 2 ATP, 2 NADH. And every time we make the ATP here, we make it in a way that is called, you ready for this? It would be useful if you retain this. Substrate level phosphorylation. Substrate level phosphorylation. All that means is, it's a fancy word. Substrate level phosphorylation. We know those 3 words, don't we? Phosphorylation means putting a phosphate onto something. Level is just, stage or way or system. And substrate is directly from the thing that the enzyme is working on. In this case, our sugar carbohydrate fragment. What it means is, when in glycolysis we make ATP, we make it by taking the phosphate directly from the sugar fragment. You're thinking to yourself, what's the big deal? Why are they even saying that? Well, it's because we're going to see a very different method in a moment. Yeah? In a moment we'll move on to see oxidative phosphorylation, and that's what happens in the mitochondrion. But for right now, this is substrate level. Yeah, a more simple form. Anything else you need to remember? Not really. Remember, this all happens in the cytoplasm. Very important. What travels to the mitochondrion? Pyruvate. So everything after pyruvate will be happening inside the mitochondrion if we are talking about aerobic conditions. And we'll talk in a moment about what happens in anaerobic conditions. Okay, so notice, no carbon is lost. 6 carbons in, 6 carbons out. In comes 1 glucose, out comes 2 pyruvate. No carbon lost, no CO2 produced. It occurs also without any oxygen input. You don't need to have oxygen present to carry out glycolysis. In our bodies, we need to have oxygen present for the later processes. Also notice, net gain of 2 ATP. If oxygen is present, we will send along NADH to a thing called the electron transport chain. We'll look at that in a moment. That's coming up. Pyruvate is the thing that is transported across the mitochondrial membranes. You can increase that to membranes because pyruvate will be transported not just across the outer membrane, but it will be transported across the MIM as well. Where will that mean? What compartment will it be in? If it's gone through the outer and the MIM, it will be in the matrix. Yeah? Wonderful. There it is in summary. 2 in, 4 out means net 2. So down here we've got 2 pyruvate, 2 ATP, 2 NADH, and 2 H+. You can disregard the protons, disregard the water here, disregard the electrons and the hydrogen ions here. You can simplify this diagram a lot. Is that okay? Will I show you again what you can cross out on your page? Get rid of 4E- and 4H+. Not interested? Get rid of the H+ here. Get rid of the H2O. And you can do likewise down here. Get rid of the 4E and the 4H+, and get rid of the H2O and the H+ here. Is that okay? And then that very simple summary is what you should begin with in any question where you're asked to talk about glycolysis. Okay, now what happens after glycolysis? Okay, well the first thing we're going to consider is anaerobic conditions. You need this word, fermentation. Fermentation is the process by which the cells attempt to recycle a product. What product are they attempting to recycle? NADH. Yeah, they're trying to get that NADH and turn it back into NAD+ to keep glycolysis going. Yeah. NADH is very useful if oxygen is around, but if oxygen is not around, it's not useful and it can become a problem. Okay, so we're going to give you two different models. One is what happens in yeasts and plants. In that situation, they produce a 2-carbon fragment, a 2-carbon alcohol, ethanol for example, and that means that they need to get rid of carbon. And so when we see plant and yeasts carrying out fermentation, we expect that there will be carbon dioxide released. And this makes the beer fizzy, yeah, and it makes the dough rise. Isn't that right? No. In animal systems, instead of converting the 3-carbon pyruvate to ethanol, the 3-carbon pyruvate is converted to the 3-carbon lactate by a redox process that regenerates NAD+. Yeah? However, having large quantities of lactate in your muscles is not good. That will bring about the pain that we feel when we get that stitch or that pain from anaerobic muscle exercise. It's unsustainable, lactic acid fermentation. Okay, so which one are we looking at here? This is, what have I got you shown so far? This is glycolysis, because it begins with glucose and ends with 2 molecules of pyruvate. Okay, we know that this gives us 2 ATP, isn't that right? Net. Yeah. And we also know that it gives us 2 NADH. Perfect, no problems. Now, if we convert this to 2-acetaldehyde, producing 2CO2, are we talking about yeast or animal cells here? Yeast. Yes. And then this eventually will be produced to ethanol. Getting from acetaldehyde to ethanol gives us another redox reaction, where the NAD+ reaction is reversed. Yeah. Is that okay? Yeah. Meaning that you have recycled your NAD+, and all you've got is a net benefit, a net gain of 2 ATP. Is that enough reason to do this process? Yes. Okay, animal cells produce the same glycolysis. They produce the same 2 ATP, the same 2 NADH, and the same 2 pyruvate. But what they have to do is they have to convert the pyruvate to lactate, which has the same effect of converting the NADH back to NAD+. Yeah, and if you look at the molecule pyruvate or pyruvic acid and lactate and lactic acid, you'll see that it's a simple redox conversion. Yeah. But lactic acid building up in your muscles is no good either. Unsustainable. Okay, that's all we're going to say about anaerobic respiration. What you need to remember is you need to remember that it must be called fermentation. You need to remember that the purpose of fermentation is to get back to NAD+. Yeah, is to get rid of that NADH. And you need to remember that plants do it by going via a 2-carbon alcohol, so CO2 is produced. But an animal cell would do it by going to a 3-carbon lactate, so no CO2 is produced. But for an animal cell, it is unsustainable. Yeah, or for human cells anyway, it's unsustainable. Now let's consider in aerobic conditions, or in an aerobe. We move on to aerobic respiration now. Glycolysis is not aerobic. occurring in the mitochondrion. And that's because pyruvate moves to the mitochondrion, doesn't it? Yeah. So that means that everything from here on out is discussed from the point of view of the mitochondrion. It's a significant improvement in efficiency over anaerobic respiration. So presumably glycolysis was going on in the very earliest of cells. After photosynthesis began in the early atmosphere and started introducing large quantities of oxygen, then some individual cells managed to use oxygen and carry out aerobic respiration, and this was quite a revolution. Because the efficiency of aerobic respiration is something like 18 times more efficient than anaerobic respiration. Yeah. So instead of making 2 glycolysis, you can make instead of making 2 ATP just using glycolysis, you can use glycolysis and aerobic respiration and create something in the region of 36 to 40 ATP for every glucose. Yeah? It is much better, yeah. And so overall, this is the equation that you should be familiar with, and the one that you would need to quote. Yeah, CO2, H2O. You can balance it out if you like, but it's not particularly important. Okay, so what does aerobic respiration consist of? It consists of 2 parts for us. The first part shall be known as the matrix reactions. Why do we call them the matrix reactions? They occur in the matrix of the mitochondrion, yeah. Is that where pyruvate goes? Yes. So is pyruvate the first substrate in the matrix reactions? Yes. Yes, it is. Yeah. Glucose becomes pyruvate in the cytosol. Pyruvate crosses the 2 mitochondrial membranes to the matrix, and the matrix reactions begin with pyruvate. Yeah. After the matrix reactions, there will be something called oxidative phosphorylation. What is being phosphorylated in oxidative phosphorylation? ADP is going to be phosphorylated, which is the end result, really. Isn't that what we're trying to do here? Yeah. What we're going to, yeah. Okay, so we know that in cellular respiration, oxygen will be consumed, water will be produced, carbon dioxide will be produced. Yeah. So during which part of aerobic respiration, matrix reactions or oxidative phosphorylation, do you think that oxygen will be consumed? Yes, during oxidative phosphorylation is when oxygen will be consumed. During which part do you think carbon dioxide will be evolved? In the matrix reactions, absolutely, yeah. That's where carbon dioxide will be produced. And where do you think water will be formed? Oxidative phosphorylation, yeah. That's exactly where water will be produced, yeah. So oxygen is going to become H2O in the oxidative phosphorylation, and ADP is going to become ATP. In the matrix reactions, pyruvate is going to become CO2. Yeah? Okay, so let's take a look at those then in some more detail. Right, this is overview again first. So here we have already covered glycolysis, and that is where glucose becomes pyruvate with the production of a modest amount of ATP and with the production of some NADH. The NADH is going to journey to the mitochondrion, and the pyruvate is going to journey to the mitochondrion. Now, these are the matrix reactions here, and most of the matrix reactions is accounted for by a thing called Krebs Cycle. In fact, some people consider Krebs Cycle and the matrix reactions to be the same thing. Is that alright? I'll try to explain this duality in a moment. Now, from the Krebs Cycle, we are also going to get a little bit of ATP created, and we're going to get lots more reduced cofactor. We're going to get lots more NADH, and also a thing called FADH2. Which is very, very similar in the way it functions to NADH. All of this NADH and FADH2 is all going to go to this electron transport chain and oxidative phosphorylation here. For our purposes, oxidative phosphorylation will do for everything. Electron transport chain and the creation of the ATP. And then using an enzyme called ATP synthase, there will be a lot of ATP created. Is this a useful diagram? You should try to envisage it in your head and try to practice drawing it on a blank sheet of paper to see if you can figure out where everything goes. All the major inputs and outputs are here, and also the stages at which everything happens. So it's a useful memory exercise to try to produce this on a blank sheet of paper, see how far you went wrong, crumple it up, start again, run this a few times, and you will get proficient at it. Okay, so let's take a look at these matrix reactions. So in order to begin the matrix reactions, we know that we need to move the entire process to the mitochondrion. Can you see the two membranes that we need to cross? Yeah, the outer membrane and the MIM. Yeah. And then you end up in what compartment? The matrix. Which compartment will you have passed through? The intermembrane space. Okay, good. Good. So we've swapped in some letters. So cover over your last picture and see if you can identify. What is A? Glycolysis. Good. What is B? Krebs cycle, or it could be matrix reactions. Yeah. What is C? Yeah. Oxidative phosphorylation or oxidative phosphorylation and electron transport. What is D? No, pyruvate. Look at pyruvate. Pyruvate is a product of glucose that crosses two membranes to move to the matrix. No, NADH and FADH are produced by glycolysis and matrix reactions and travel that way. What gets broken down at the end? Nope. You're right. ADP does need to enter in order to produce ATP, but I think D and E are matched together. They're part of the same process. What comes in? Oxygen. Yes, and oxygen, through this process occurring at C, will be used to form water. Yes, very good. Okay, is there any other letters we need to cover? No. Okay, so on we go. Now, sometimes in your exam, they will look at the matrix reactions, which look like this, and they will say, these are link reactions or preparatory steps, or I've even heard them called chemical grooming, which to me sounds creepy. Yes? And then down here they will call this one Krebs Cycle or the TCA cycle, tricarboxyl, no, not TCA, citric acid cycle. Is that alright? Sometimes they will say this is Krebs cycle. Yes? What that means is, sometimes they'll ask you a question with, name the carbohydrate product that is fed into Krebs cycle, which leaves you guys in a difficult position. Do they mean this product? Or do they mean this product? So what do you do when faced with ambiguity in your exam? What do you do? Absolutely, you explain the problem. You say, if Krebs cycle is presumed to be beginning at the link reactions, then the first molecule is pyruvate. Yeah? However, after the link reactions fed directly into the cyclical reactions of Krebs cycle is another one, which you've probably met before. This guy over here, acetyl-CoA. Yeah. So let's look at them separately for a moment. I prefer to call these the link reactions, to call this Krebs cycle, and overall I prefer to call this matrix reactions, which includes the link and Krebs cycle. Is that okay? But unfortunately, you need to bear both of these in your mind, because they could frame the question either way. You need to be prepared. Okay, so what's happening in these link reactions? Well, 1, 2, 3, it's laid out in this diagram here. Number 1. It's more than that, really. Transport has occurred, okay? So now we're in the matrix. Yeah? Are there any parts, 1, 2, and 3, that occur outside the matrix? No. So, we must remember always, everything that happens in the matrix reactions is already inside the matrix of the mitochondrion. I don't want to hear anything about acetyl-CoA in the cytosol. Don't do it. Okay, so what happens? Well, number 1, we're going to have the loss of a carbon molecule, carbon atom. So CO2 is going to be reduced. We'll move from being a 3C compound to being a 2C compound. Simple enough. Carbon dioxide. This is the first time we've lost carbon dioxide, isn't it? Is it? Have we lost carbon dioxide before? No. We brought 6 into glycolysis and 6 came back out. Okay. Second thing that happens. What kind of reaction is number 2? Redox. Again, it's another oxidization. So we have oxidized the carbon fragment again. Just like we've done during glycolysis. We did it in glycolysis on one occasion, resulting in 2 different NADH, one from each of the 3 carbon fragments. And now we're doing it again. Is that okay, Fatima? You look worried. We've oxidized it. A very simple oxidation step. And then what we're going to do is we're going to temporarily complex it with another coenzyme. Don't worry about it. All we're going to do is attach it to this thing, move it into Krebs cycle, take the other thing back off again. So essentially it just means a way of shepherding it into the cyclical reactions. We're temporarily going to attach it to a thing called coenzyme A. And that ends up with the product acetyl coenzyme A, which is another very important metabolic intermediate. It interfaces with many other metabolic pathways. We only care about one metabolic pathway here though, and that's aerobic cellular respiration. So acetyl-CoA is very important for going on and creating fats, etc. But we're just going to talk about it as a catabolic breakdown product that's being fed into the Krebs cycle. So, what molecule goes into Krebs cycle? Pyruvate, if you consider Krebs cycle like we do on the right. But if you consider Krebs cycle like I prefer it, it's acetyl-CoA. Yeah, you have to bear in mind that the question could be framing it in either way. Yeah. It's not right to have ambiguous questions like that, but if it occurs, I want you guys to be prepared for it, and I want you to deal with it, collect your marks for sure. Yeah. All right. So after these preparatory steps or link reactions, we have a thing called acetyl-CoA. How many carbons are in this fragment? 2. Yeah. Okay. So this is the Krebs cycle. There's Mr. Krebs. It's not a possessive. It's not the cycle that belongs to Kreb. It's the cycle that has been named after Mr. Krebs. Do you know what I mean? Yeah. So you capitalize the K, the same as the Golgi buddy. Okay. This is a step, this is a process that is catabolic. At every stage, the molecules are becoming bigger or smaller. Are they becoming more complex or are they becoming simpler? Simpler. Is it progressively being oxidized or reduced? Oxidized. It's losing all the time, isn't it? Yeah. This is quite normal for catabolism. It's what we saw in glycolysis, isn't it? Pyruvate, 2-pyruvate are smaller, simpler molecules than glucose, and they were losing all the time, weren't they? They were losing mass, they were losing protons, they were losing electrons, they were losing free energy. Yeah? Okay. What you need to know about the entire matrix reactions is contained on this slide. Yeah, so don't worry about this guy, but it's a very complicated series of enzymes, intermediates, multiple steps occurring at different stages. We don't need this stuff. We don't need this stuff at all. The only thing that would be useful, if you want to show off in your exam, is learning off the name of this, this, and this. The way you start a cyclical process is to bring in a small addition. So we have oxaloacetate. Oxaloacetate, how many carbons has it got? 1, 2, 3, 4. Yeah. What we do is we take oxaloacetate and we combine it with acetyl-CoA, which has 2 carbons, producing citrate, or citric acid, which has 6. And then as citrate makes its way through all these steps all the way around, it will lose how many carbons? 2. It will also be oxidized several times, losing protons, electrons, and free energy. And after it's lost protons, electrons, free energy, and carbon, it will be back as oxaloacetate. And you can bring in another acetyl-CoA. So every time Krebs cycle turns around, 2 more carbons came in the form of acetyl-CoA, bringing electrons, protons, and free energy. And, 2 seconds, and then as they go around, we will lose carbon twice as CO2, and we will lose electrons, protons, and free energy, creating NADH, and we're back to oxaloacetate again. So the last product, if you will, becomes the first substrate. Yeah, that's a cyclical process, a cyclical pathway. Yes, Vivian.
Yeah, so point that it happens twice because there's 2 pyruvate.
Not it happens twice?
No, I mean like
A single journey involves... different point. Yes, you're absolutely right. So one single turn of Krebs cycle takes in 1 acetyl-CoA. Yeah. But 1 glucose, how many acetyl-CoA does that produce? 2. You're absolutely right, Vivian. You keep an eye on the numbers. Because at the end of it, we will have brought in 2 pyruvate. That will have both gone through the link reactions, producing 2 acetyl-CoA. So Krebs cycle turns twice for every glucose that comes in. Yeah? At the end of it, what are the outputs? Here they are summarized. We brought in 2 pyruvate. Those 2 pyruvate will produce 2 NADH in the link reactions. They will produce 2 acetyl-CoA, which go into Kreb cycle. And those going around through Kreb cycle will produce 4 CO2, 6 NADH, and 2 of a thing called FADH2, which is exactly the same as NADH. We'll look at it in a moment. The very same. It does the same job. And it will also produce 2 ATP. Yeah. Again, a small amount of ATP. But what we have accumulated a whole bunch of is CO2 that needs to be released as a waste material. And we have also accumulated a whole bunch of reduced coenzyme. NADH and FADH2. Yeah? Which is bad news if you don't have oxygen. Would Krebs cycle run if you didn't have oxygen? It's not directly requiring of oxygen. However, it produces such a vast quantity of NADH that unless you have a steady supply of oxygen to allow oxidative phosphorylation to run, to allow oxygen to take the electrons from NADH, you wouldn't be able to run Krebs cycle for anything less than a fraction of a second.
But like in theory, Krebs cycle would run without oxygen. It's
only if it was coupled with another process occurring simultaneously to remove the NADH. Yeah. So in the same way that without ATP, endergonic reactions won't run, without oxygen, Krebs cycle won't run. Yeah. Is that okay? We'll pick that up the next day. Not the next day, later on this afternoon. Yeah. Wonderful.